Hello and welcome to your Chapter 15 video lecture series. In this chapter, we're going to talk about genes and proteins, and we will start with section 15.1 on the genetic code. Our learning objectives here are to explain the central dogma of DNA protein synthesis and describe the genetic code and how the nucleotide sequence prescribes the amino acid and the protein sequence. So let's first revisit proteins. We know that the monomer for proteins will be amino acids, and there are going to be 20 of the most commonly occurring amino acids in these compounds over here. And we can think of these as the protein alphabet. And each is going to have a different type of chemistry. Some are going to be acidic, some will be very basic, some are polar, some are nonpolar, and there are different structural constraints as well. So let's talk about that central dogma. Now we know that DNA is going to direct and regulate the construction of our proteins that we need for cell growth and reproduction in a particular cellular environment. And so we're going to start off by first replicating our DNA, which we talked about in chapter 14. And then that DNA is going to be utilized to go through a process called transcription. Transcription is going to essentially take a gene that is composed of DNA and read it or transcribe it to produce what's known as messenger RNA or mRNA. And then we take that mRNA and go through a process called translation, where we are able to take that information from the mRNA and translate it into a polypeptide, which then can fold and become a protein. And to do this, we're going to utilize our ribosomes. And you can see that pictured over here, where we're going to have a molecule that we'll learn about in the last section known as tRNA that will come in, and we will start linking the amino acids that it carries on over. Now you may be wondering, why do we even bother transcribing DNA into mRNA? Why don't we just read the DNA and create a protein? But this temporary molecule is created because we want to keep DNA intact and protected. And so we kind of make a copy of that, a temporary copy, and then utilize the copy to create our protein so we don't damage our DNA. And by going through this process, we are able to give our genes expression. And this process is known as our central dogma of DNA encoding our RNA and then the RNA encoding protein. So let's talk about RNA transcription first. Like we said, we are going to take information that's encoded within our DNA, this would be serving as our template strand, and then transcribe it into an mRNA strand or a messenger RNA strand. And this is just going to be one strand of information, whereas we know our DNA is a double helix. And the other difference we'll see here is that with DNA, we're going to utilize the nucleotide thymine, but mRNA or RNA in general cannot use thymine. Instead, we are going to utilize uracil in place of thymine. So note, anytime we have adenine on the DNA template strand, we are going to pair that up with uracil. Next, let's talk about the genetic code. It is a degenerate and universal code. So first, let's think back to our central dogma, where we mentioned that that mRNA is going to be translated into a protein. And in order to do that, we have to convert a nucleotide-based genetic information into a language of amino acids. So you can see this table over on the right-hand side. You also may notice that we've got 3 letters representing our nucleotides set together within these squares. And what this represents is what we call a triplet codon, where we are going to take that first letter, let's say I'm going to randomly choose one, C, and then our second letter, let's also say is a C. So now we're going to look to this and we drop down to this box, and then our last letter can be an A. Now it seems that that third letter doesn't matter so much because all of these are going to code for the amino acid proline. And if we consider all of the letters that could be combined, we have a total of 64 possible combinations, which will help set up these 16 blocks of codons that are really specified mostly with that first and second letter. But you can see in certain instances that third letter is going to be important. For instance, in this block, we see that if there's a U or a C at the end, that is going to code for histidine, and if we change it to A or G, then it is going to code for glycine. Whereas others, like leucine over here, is going to be able to have the 3 letter mark not make a difference, but also have some extra triplet codons also code for it. So we have a total of 6 codons that could code for leucine. And this is what we mean by the term degeneracy, or the genetic code being degenerate, because it means that an amino acid can be encoded by more than one nucleotide triplet, and our normal proteins can't be produced when only 1 or 2 nucleotides are used. And it seems that it was created this way so that if we did have a mutation take place where we get a nucleotide substitution in this third letter area, that it would still code for that same amino acid. And so that would not really affect the protein, and it would still be functional. Now, this is important when we think about what happens when we get an insertion of 1 or 2 nucleotides. This can completely change the reading frame. If you look at the very top over here, we could see that we've got triplet codons. And if we were to accidentally insert some other nucleotides in here, it shifts that frame so that instead of coding for these amino acids listed here, you can see that there is different ones listed right after that insertion has been made. Now here we've only had an insertion of 2 nucleotides, but if we were to have had 3 nucleotides added, it would add in a different amino acid, but the rest of the protein would keep its integrity because they are going to read the 3 nucleotides together that would code for amino acids. And something that's really incredible about our genetic code is that we see that there's this relationship between our mRNA codon that we have and its corresponding amino acid. And this is nearly universal. Almost all species use it. So it doesn't matter if protein synthesis is happening within a plant or a monkey, we're going to utilize the same tools to code for these same amino acids. And to begin protein synthesis, we're going to need what's called a start codon, which is AUG. That actually is going to code for a specific amino acid known as methionine. And like I said, this is going to initiate translation. Essentially, we're taking this reading frame and it's beginning with AUG, which codes for methionine. And this would happen near the 5' end of our mRNA strand. Now, once we create that protein, we are then going to run into one of 3 stop or nonsense codons. And you could see these listed over here. We've got UAA, UAG, and UGA. That when that ribosome is moving along the strand reading it, and we see that stop codon, it terminates protein synthesis and will release the peptide. Now, just a quick note, we are going to skip section 15.2 on prokaryotic transcription because you will cover this in a microbiology course.